Jalan-jalan dulu ya, Ma. Oli mana?
I'm sorry, we are still waiting for Din Ali. He will be join us soon.
Iya. Yang udah mungkin di-admit dulu, enggak, Mbak?
Iya, kenapa Dok?
Yang udah waiting mungkin di-admit dulu kali enggak? Atau nunggu Pak Dekan?
Oh, langsung aja kita admit ya, Dok?
Admit aja terus mute semua aja kan, tunggu Pak Dekan.
Iya, oke, baik. Okay, Prof. Ali is still joining. Thank you. Good afternoon. You may begin. Din Ali, I'm sorry, can you hear us?
Yeah.
Okay.
Okay.
Okay. Good afternoon, everybody. Welcome and thank you for joining our lecture. Before we start, I would like to invite Din Ali to deliver opening speech. Please, Din Ali, the time is yours.
Okay. Assalamualaikum warahmatullahi wabarakatuh. Yes, distinguished our guest speakers, Professor Bruno Guigas from Leiden University Medical Center, and also we have a speaker from Faculty of Medicine, Dr. Dicky, and also Dr. Farid, and our moderator for this afternoon, Professor Dr. Pradana Soewondo, internist consultant of endocrine metabolic diabetes. I'm Tahapary as the Dean of Faculty of Medicine of Indonesia, would like to extend my warmest welcome to all participants, especially today's guest lecturer, Professor Bruno Guigas, PhD. It is such an honor for us to have your presence within the online space. Today's lecture will bring up to the topic of obesity and the type 2 diabetes, new insight, which will be delivered by our distinguished colleagues, Professor Bruno, and as well as from our staff, Dr. Dicky and also Dr. Farid. And I think this is the important topics now, not only in this pandemic era, because you know that the many people just sitting on their laptop and then to reduce their activity, and it will be risk factor for obesity and also diabetes. And I hope that all participants can actively engage within every sessions and have fruitful discussions, which will be led by our senior professor, as I mentioned, Professor Dr. Pradana. Furthermore, through these programs and many more to come, I also wish that this will enhance the collaborations between Factor Medicine Indonesia with Leiden University Medical Center in terms of joint research, publications, and also lecturers or webinars. Lastly, I would also like to give my appreciation to all committee members who have taken part in today's guest lecture. And I also would like to give my sincere gratitude again to Professor Bruno that has given his time to share his knowledge with us. Okay, finally, I hope that everyone who gathers here can take benefit from today's discussion. Please, Professor Pradana, can continue this session. Thank you for your attention. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Professor Ari Faryal, for the opportunity, and it is a great honor for me to lead this Indonesia and one from this Professor Bruno and this section is a senior lecturer from the Department of Economics, Division and
Thank you, Professor Pradana, for the very kind introductions. Good afternoon, distinguished guests, also Honorable Professor Syam, as the Dean of the Faculty of Medicine Universitas Indonesia, as well as to Professor Bruno Guigas from LUMC. So I would like to share my screen. So the title of my talk for this evening is actually about the burden of metabolic diseases in Indonesia, an even more critical issue during COVID-19. So this would be only a general overview to set the stage for the next two speakers. So here, if you look into the metabolic disease in Indonesia, there is an alarming increase in the prevalence. If you look based on the latest basic health research, the prevalence of obesity pre-diabetes, dyslipidemia, and hypertensions, almost all of them were reaching more than 30%. And even more about the prevalence of pre-diabetes. It already reached 45% in rural and 35% in urban area. And if you look into the prevalence of type 2 diabetes, it's already reached about 10% of adult populations. And what is challenging as well is actually about the proportions of early onset diabetes among those aged between 15 and 45%, which contribute about 25% to all diabetic patients. So we need to screen at the earliest and also identification of a high-risk group and also perform a prompt intervention starting at the community level. So the higher metabolic disease has also impacted the high morbidity and mortality associated with cardiorenal complications. It is also associated with worse outcome infectious disease, for example, tuberculosis, which is endemic in Indonesia, and recently COVID-19. And if we discuss about COVID-19 pandemic, the relation between metabolic diseases and COVID-19 can be bidirectional. There are several factors. The first one is the high prevalence of metabolic disease in Indonesia, including those in younger age, and also closer intergenerational contact and limited healthcare capacity might increase the risk for severe infections. That will necessitate the importance of COVID-19 prevention in this high-risk group. But on the other hand, actually, COVID-19 drives disruption in the chronic care of metabolic disease, forcing faster adoption of digital health, for example, telemedicine. So if you look into this problem, actually, the increasing prevalence, also the increasing numbers, especially in younger age and also in some part of the rural area, we need the responsive changes in policy and as well as on research. Will be later on discussed by the next two speakers. So if you look into Indonesia, we have a really diverse geographical and biodiversity variations. For example, if you compare between rural area of Flores and also urban Jakarta, you see there is striking changes in terms of environmental as well as lifestyle of those people living in rural and in urban area. So these differences in lifestyle, as well as break with natural environment and biodiversity, appear to be associated with changes in the immune system, resulting in various adverse health effects. So although genetics can have a major impacts on the immune system and disease susceptibility, the contribution of environmental factors is thought to be substantial. So our previous study in collaboration with LUMC, we have two centers, one in Nangapanda area, which is a rural area, and the other one is in Jakarta. We have collaboration with LUMC, also with IMERI. We have already started to learn a framework of a new pathogenesis in terms of development of type 2 diabetes. Not only about lifestyle factors, but the loss of diversity across the changes with urbanizations. For example, with the deworming program, less health infections, although improved hygiene, will lead to increased obesity and also increased inflammation, which will then lead to insulin resistance, which is actually the main pathogenesis for the development of pre-diabetes, type 2 diabetes, hypertension, dyslipidemia, as well as cardiovascular disease, and is associated with increasing risk of infections. Our previous study have shown that the changes between urban area in Caucasians, also in rural area of Indonesia, which is endemic for helminth, there is a really changes in terms of activations of immune cell. Those living in rural area have a more anti-inflammatory phenotype compared to those living in urban area. So in a general level, you can see there's a difference in terms of environment and biodiversity. And in terms of at the human level, you can see there's a difference in terms of inflammations, increase in adipose tissue among those with insulin resistance, which will later on be discussed by Dr. Farid. And what we want to discuss later on with Professor Bruno is actually about the immunometabolism, the changes that occur intracellular metabolic pathway in immune cells during activations. So the change is not only on the level of the environment, as well as on at the human level, but there's also change in the cellular level in the metabolism pathway. I think that would be all for my talk. I would like to give the floor for Professor Pradana Soewondo. Thank you very much for your kind attention.
Okay, thank you very much, Dr. Dicky Tahapary, for your nice talk. It's already shared the data from Indonesia, especially from the urban to rural transition. Yeah, I think this is a very important study that already done by Dr. Dicky Tahapary. So the second speaker will be Dr. Farid. Dr. Farid is a PhD candidate from the LUMC under supervision of, among others, Professor Bruno. So he will talk about the obesity, especially from the youngest generation. So please, Dr. Farid, for your presentation.
Thank you, Professor Pradana, for the kind introductions. Today, I'm going to share. Let me share my screen. And thank you also for Professor Ari and the committee for letting me to share my work for now regarding with my PhD study or with the other research together with Professor Pradana and also with Dr. Dicky. Actually, Dr. Dicky already mentioned quite a lot about what I'm going to talk today, so it's quite not really hard. I hope it's not really hard topic to follow. So as an introduction, as we all know that Dr. Dicky mentioned that Indonesia now is facing a double burden of disease, a transition from the still high prevalence of communicable diseases like tuberculosis, dengue, helminth infection or parasites, to an increase of non-communicable diseases like obesity, type 2 diabetes, or cardiovascular diseases. Here from the basic National Health Survey in 2018, we could see that the prevalence of overweight, obesity, and central obesity is steadily increased from year to year. Also, the prevalence of diabetes as well. So come up the questions, what are the causes of this phenomenon? As a developing country, Indonesia undergo a rapid socio-economic development. We have an increase of GDP, and those rapid socio-economic development will translate into an increase or rapid rate of urbanization. Here, urbanization is not only translate into a migration of people from rural to urban, but also the adaptation of urban lifestyle in rural area also could be called as urbanization. Sorry, you can hear my voice clearly?
Yeah, yeah, it's clear. It's clear.
Okay, thank you. Between urban and rural, there are several characteristics that really differs between those two environment or population. In urban, the population or the individual is less physical activity with higher fat and higher calorie in their diet, and also with higher stress level and also pollutions. Meanwhile, in rural, the people tend to be more active with lower calories and fat and more diet in their more fiber in their diet, and also with relatively more pathogen exposures in some part of rural like parasites or helminths, and also don't mention about the farm environment exposures in part of rural. And these differences are subjected to differences in the prevalence of obesity and type 2 diabetes. the study that Dicky and I conducted previous years, that living in urban is associated with worse adiposity profiles. We recruit 86 male subjects and 40 from rural and 49 male subjects from urban with same ethnicity, Flores ethnicity, and we measure the adiposity, and we see that every 1 year increase of time that's spent in urban area was associated with significant increase of BMI and waist circumference. And previously, Dicky already mentioned a lot about the young adult population or early adulthood populations as a definition in some paper or some publication. Young adult is defined as people with age 18 to 25 years old, but other publication also mention until 30 years old individual. But as we all know, a recent publication from JAMA 2018-2017 mentioned that this population is a vulnerable population, but often neglected. Why they are vulnerable? Because they are experience a lot of significant life transitions, like living independent from their parents, and also urbanization that we're going to talk more after this, for following or pursuing higher education, and also for finding a full or part-time job. And then those life transitions cause them to expose toward a new environment, and then a change also in lifestyle. These changes cause them to make risk-taking behaviors that makes them prone to excessive weight gain and also increased prevalence of obesity. Sorry, it seems that my voice is echoing. It's still echoing or not?
No, it's fine.
No, it's okay.
And the health behavior established during this period often persists into life and potentially affect their offspring. So it will affect in the very long, prolonged period, not only to this individual of this young adult, also have the potential to affect the offspring or their children itself. Then, in 2017, their association of weight gain from early to middle adulthood showed that there is an increase of individual health outcome with the each per 5 kilogram weight gain. We could see that the type 2 tension and also cardiovascular diseases is the prevalence is increases. For studying this young adult as a very interesting populations and very vulnerable population, we further conduct research that recruit freshmen or first year undergraduate student of University of Indonesia as a prospective cohort study. We divide them into two groups. The first one is the urban group, the subject or the student that born and live in urban or metropolitan area, and the rural group, that is the student that born and live in rural area that recently migrate to Jakarta, and there's no history of previously ever stayed consecutively for 6 months in urban area. We follow up these two groups for 2 years and with yearly follow-up. And then for the baseline, we could see here that the result is in concordance with previous study. We could see here that the BMI is higher in the urban compared to rural, and also with the waist circumference. And we could see here also the proportion of the subject with overweight and obesity is much, much higher, as well as the central obesity is much, much higher in urban compared to rural. So we could conclude that the urban Indonesian young adults have worse adiposity profiles compared to rural. And it seems that these differences could be partially explained by the higher intake of calories, fat, and also protein. In the case of our study, that you could see here that those intake of calories, fat, and protein are higher in urban compared to rural. This data showed also that the cholesterol level, like the total cholesterol, LDL cholesterol or bad cholesterol, and triglyceride levels also higher in urban compared to rural. And the insulin resistance that's stated by HOMA-IR are also higher in urban compared to rural, even after we adjust for the BMI. After we see those data, that it seems that rural are with the better metabolic profiles compared to urban, but we want to know further how are they respond to we stimulate like the short-term high-fat, high-calorie diet, like the usually observed in urban population or urban environment, and we see that after we give the subject 1,500 calories on top of their regular diet for 5 days, we could see that the insulin resistance both in urban subjects and in rural subjects all increase. But it seems that the increase is more pronounced in the rural subject, although it's not significant. So living in rural, although we could see that they have better metabolic profile, but does not protect them from the metabolic changes. In this case is the increase of insulin resistance after short-term high-fat, high-calorie diet. So furthermore, like I already mentioned before about the prospective cohort study of the young adult population, in this case is the first year undergraduate student of University Indonesia, we could follow up them after 1 year. Unfortunately, because of this COVID-19 pandemic, the second year follow-up we could not perform. And we could see here that after 1 year living in urban environment, the increase of BMI is significantly higher in rural compared to urban subject. It's almost twice higher. And this contributed by the higher increase of calories intake and also protein intake in rural. Although for calories it's not statistically significant, but carbohydrate intake also seems like higher in rural compared to urban after 1 year living in urban environment. So this finding also conclude that the rural subjects are more prone to metabolic change, metabolic profile changes if they are exposed to urban lifestyle. So the questions is, what is the pathophysiology of these metabolic changes that follow urbanizations? We could see that what I showed before, the less physical activity and also high fat, high calories diet will induce a positive energy balance, and this positive energy balance will be stored in the body as a body fat. With the increased body fat, then it will also increase the adiposity and insulin resistance. This pathway usually considered as a traditional pathway or conservative pathway, but from relatively new studies or previous research in this last decade, we could see also that this pathway also involve we call the gut ecosystem, gut microbiota and gut permeability that also already mentioned by Dr. Dicky before, related with the biodiversity between rural and urban. When they moved from rural to urban, they lost their biodiversity, including the gut microbiota and the gut microbiome. And also, not to mention about the pathogen exposures, in rural or certain rural, we often see the higher prevalence of helminth infections, but after they move to urban, they lost their pathogen exposures. Why? This is important. We will see later after in this upcoming slide. These changes in the gut ecosystem, the gut microbiome, will affect the immune system, and there are a lot of publications that state that the gut microbiome all have a really close connection with our immune system. And with the changes in the immune system, this could also change the adiposity and also especially insulin resistance. Here we could see that from several studies state that showed that obese and type 2 diabetes subjects exhibit a decreased biodiversity and richness of the gut microbiota, and also decrease the relative abundance of certain beneficial bacterial populations such as butyrate producing bacteria. And this similar pattern are also observed in urbanization. So we could say that urbanization might promote changes in adiposity and insulin resistance via changes in the gut microbiome. Here, the dysbiosis of the gut microbiome could be affected by diet, but also with other factors like age, lifestyle, antibiotic usage, genes itself. And this influence the host gene expression and energy harvest, and the chronic inflammation, and induce an obesity or other metabolic diseases like diabetes. If we talk about urbanization, we could not forget about this high-fat diet or high-calories diet. And this high-fat diet could induce an increase in the gut permeability by modulation of the gut microbiota composition and also the reduction of the tight junction proteins. And this reduction of these tight junction proteins will cause a metabolic endotoxemia, where the lipopolysaccharide from the gram-negative bacteria in the intestinal lumen could easily goes into the circulating system and activate the toll-like receptors on the immune cells and cause the low-grade chronic inflammation that eventually will induce an insulin resistance. Interestingly, this pathogenesis of insulin resistance and type 2 diabetes, there's a shift of paradigm after previously in the early 2000s, the Fronso called the triumvirate, the pathogenesis of type 2 diabetes that involve only 3 organs, pancreas, liver, and also muscles. In 2009, he proposed 8 organs, and Schwartz in 2016, he added more 3 organs, and one of them is the colon or gut microbiome with the perspective of the interventions. Talking more about the pathogen exposures, I've mentioned before about the parasitic infection, especially soil-transmitted helminth. The study by Wirya in 2015 showed that here, interestingly, that soil-transmitted helminth is associated with lower insulin resistance. Here we could see that more species and the lower the insulin resistance. And study by Dr. Dicky that continues this previous study by Wirya, that eradication of those helminth infection with anti-helminthic treatment resulted in the increase of BMI and insulin resistance. And this effect is more greater in the subject with multiple infections. And those one, I've told you about the pathophysiology regarding the gut ecosystem, and now the pollutions. Pollutions from this study by Young in 2000, just recently in 2020, showed that with the increase of particulate matters, 10 gram per 10 gram increase of particulate matters, it will increase the incidence and also the prevalence of type 2 diabetes. The particulate matter could be inhaled by individual or by people, and they could go to the systemic circulation, and they activate the immune system, and they cause low-grade chronic inflammations and also insulin resistance. And pollutant like the particulate matter also considered as endocrine disruptors. So, ladies and gentlemen, we come to my last slide. As a summary, urbanization is associated with an unfavorable metabolic health profiles, and the metabolic changes related to urbanization could contribute to the pathogenesis of obesity and type 2 diabetes. And the urbanization that we see in the young adult population, in this case is the student of University of Indonesia, might potentially associated with metabolic health deterioration, especially in the long term. And early detection and prevention in the young adult population is really needed to prevent excessive weight gain and obesity in the long term, and it might contribute to the decreased prevalence of metabolic diseases. Thank you for your attention. I would like thanks to all of my supervisor, my senior, my colleagues in University of Indonesia, in LUMC, and also in IMERI. Thank you.
Yeah, thank you very much for Dr. Farid Kurniawan. It is a very interesting lecture, and there are nice data related to the transition from the rural to urban on health. So it is very interesting, and I'm sure that later on Professor Bruno will explain more detail about the pathophysiology of the this mechanism. And then I will invite Professor Bruno Guigas, PhD from LUMC, to talk about the immunometabolism in obesity and type 2 diabetes. So before you start your lecture, may I read your curriculum vitae first? So Dr. Bruno Guigas is a molecular biologist with a broad expertise in the pathophysiology of metabolic disorder. His research group at the Department of Parasitology of the Leiden University Medical Center is driving project within the emerging of the immunometabolism, aiming to identify the new drug-able molecule and or molecular target involved in the immune regulation of metabolic homeostasis and in the context of obesity and type 2 diabetes. So he has a co-author of more than 85 peer-reviewed publications and is a scientific advisor for various funding agencies and biotech companies. So please, Professor Bruno, it is time for yours.
Thank you very much. I will just share my screen now. Hoping it's going to work. Okay.
Yeah, it's okay. Yeah.
Great. So it's a great honor and a pleasure to be with you today. So I'd like to thanks the Dean for the kind invitations, and also Professor Pradana for his nice introductions. It's also a pleasure to see my friends and colleagues, Professor Dicky and Dr. Dicky and Dr. Farid. So today I'm going to talk about immunometabolism, indeed, which is a new emerging field that has provides a new light in some of the mechanism involved in the development of insulin resistance and type 2 diabetes. So my talk will be divided in two parts. I will give a very brief overview of how obesity might trigger a so-called meta-inflammations, which is a chronic low-grade inflammations and how it plays a key role in the development of insulin resistance and type 2 diabetes. And then I will move to actually my current research line, which is studying how helminths, which are parasitic worms, can actually impact the immune system of the host and eventually modify the metabolic homeostasis. So you all know that the world is growing obese. So this is the map of the BMI at the worldwide levels, and you see that there are a lot of very red colors appearing, mostly in the Western countries, but this is also important, and I think Dr. Dicky has already introduced this, that obesity is also rising in Indonesia and in Asian countries. So this is not something that is only limited to the Western style. It's present and growing everywhere. And the development of obesity, together with the development of insulin resistance and the appearance of dyslipidemia, so an impairment in lipid homeostasis, are three of the four metabolic dysfunctions that are a component of a so-called metabolic syndromes. And as soon as you're starting to have metabolic syndrome or being pre-diabetic, you have an increased risk of developing type 2 diabetes and also associated disease like cancer and cardiovascular disease. And this is just one map of also of the total number of diabetic people in the world. So it's include both type 1 and type 2 diabetes, but I mean 95% of the diabetes are actually type 2 diabetes. And what you can see here is that due to the high number of inhabitants in Indonesia, Indonesia is marked as a mild red color. So that really means that there is a high number of diabetic people in Indonesia. So it's a really important health issue there. And also a significant number of these people are actually undiagnosed. And Dr. Farid has introduced also the concept of these rural-urban transitions. And what is important is also there is a constant rise in the number of people suffering from type 2 diabetes, because there is an increased urbanization rate in Indonesia. So, but what is the mechanism underlying the development of insulin resistance and type 2 diabetes? So during the last 10-15 years, there is a new concept that has emerged, which is actually the contribution of meta-inflammations to the development of insulin resistance and type 2 diabetes. And the meta-inflammation is actually due to change in the immune cell compositions in the metabolic organs. So in every organs involved in the regulation of metabolic homeostasis, here there is 3 of these organs: skeletal muscles, adipose tissue, and the liver, but I might have added the pancreas, the intestines, even the brain. There are a bunch of immune cells that are populating this tissue. And during the development of obesity, mostly due to the fact that there is a high influx of fats and lipids and bad lipids, there is a shift in the composition in these immune cells in this tissue, leading to a pro-inflammatory environment. And this pro-inflammatory environment will lead to a tissue-specific insulin resistance, and I'm going to explain you how it goes. And this tissue-specific insulin resistance later on results in a systemic insulin resistance, and when you start to become insulin resistant, a couple of years later, you have increased risk of developing type 2 diabetes. So I will give you some examples on what is known currently in the mechanism associated with this immune-mediated regulations of insulin sensitivity. So here, I'll take as an example what's happening in the white adipose tissue. And there it is pretty well established now that there is a bunch of immune cells that are populating this healthy tissue that are mostly belonging to the type 2 immune cells. And it has been shown mostly in rodents, but that has been confirmed also in humans, that there is a network of type 2 immune cells that are maintaining insulin sensitivity. And it is believed that actually both ELC2s and Th2 cells are secreting prototypical type 2 cytokines like EL4, EL5, EL13s. And these cytokines are recruiting and maintaining eosinophils in the adipose tissue. And eosinophils has been shown to be one of the main producers of EL4. And therefore, EL4 secreted by eosinophils are actually polarizing the macrophage, the tissue-resident macrophages, toward alternatively activated states, a sort of anti-inflammatory states, mostly through increase in STAT6 signaling inside these macrophages, and this STAT6 signaling is triggered by the binding of EL4 to the EL4 receptor present on the macrophage. And all the macrophage, all these macrophages polarized toward these anti-inflammatory states are maintaining the insulin sensitivity in the tissue. This is still not very well known. I mean, people believe that that might be through secretion of some specific cytokines like EL10, because that's not very strongly established, or this macrophage can also promote certain type of protective immune cells like regulatory T cells that might dampen the inflammations. But what is sure is that even if we don't know how these macrophages are maintaining, contributing to the metabolic health in the adipose tissue, what is well known is that during the development of obesity, the population of immune cells are changing drastically. So we are basically losing these type 2 immune cells, and there is recruitment and activations of some pro-inflammatory immune cells. So NK cells, Th1 cells, Th17 cells are activated. How it occurs, this is also a bit difficult to understand now, but what is sure is that at least part of the mechanism is driven by the expenditure of adipocytes. So the adipocyte, the lipid cells are growing and being inflamed. They are secreting chemokine and cytokines, and these chemokine and cytokines are released from the adipose tissue, and they are promoting the recruitment of monocytes that are, once they are entering this pro-inflammatory milieu, these are polarized toward pro-inflammatory macrophages. And these pro-inflammatory macrophages are producing pro-inflammatory cytokines that are directly interacting negatively with the insulin signaling pathways, and that's induce insulin resistance. So this is occurring in the adipose tissue, but this is also occurring in the liver. I'm not going to describe that in details, but that's more or less the same concept. When there is a large influx of lipids in the liver, the Kupffer cells, which are the resident macrophages, are activated, and they are secreting some factors, chemokines, cytokines like MCP1, that are also contributing to recruiting newly activated pro-inflammatory cells, and therefore promoting insulin resistance. And other type of cells like neutrophils or CD8 T cells are also contributing to this phenomena, and that's all at the end induce insulin resistance also in the liver. And I could also explain what's happening in the brains, in the pancreas, but altogether, these tissue-specific inflammations are driving insulin resistance, and later on, increasing the risk of developing type 2 diabetes. So let's move to the helminths, and how helminths are actually coming into this piece. So as I just mentioned, I mean, in healthy tissue, a bunch of immune cells are mostly there and maintaining insulin sensitivity. So logically, I mean, you would say, well, I mean, if we move this pro-inflammatory type 1 immunity environment back to type 2 immunity, that would maybe restore metabolic homeostasis in people that are insulin resistant. And the strongest inducer of type 2 immunity are actually parasitic worms. And helminths are populating a lot of different countries. So this is marked in red, and including in Indonesia, which is, as you know, endemic countries for helminths. And helminths are masters in modulating immune systems. Here you can see this is a scheme depicting actually the impacts of helminth infections on immune response. And as soon as the cercaria are penetrating the skin of the host, there is a very mild type 1 immune response that is characterized by an increase in pro-inflammatory macrophage and Th1 cells, but this is rapidly going down, and when the worms are pairing inside the host and start releasing eggs, this is associated with a very strong type 2 immune response that is characterized, as depicted here, by the classical type 2 immune cells, alternatively activated macrophages, eosinophils, Th2 cells, and also some regulatory cells like B cells and T cells, and Treg, sorry, that Breg and Treg that are dampening this type 2 immune response. So, and it has been shown actually that that people with helminth infections are actually in a way protected against type 2 diabetes, or have a better whole body insulin sensitivity. So here are a map of the different field study that has been conducted, studying the impact of different type of helminths on whole body metabolic homeostasis. And one of it, as Dicky has already introduced, is a crucial and seminal studies performed in collaborations with LUMC, so performed in Indonesia at Flores Islands in collaboration with Indonesia, with LUMC, sorry, and has shown that people infected with helminths, salt-transmitted helminths, have a better OMA-ER, which is an index for whole body insulin sensitivity. And interestingly, another work that has been driven by Dr. Dicky showed that if you deworm these people, then suddenly, I mean, their metabolic profile are worsening. So suggesting that actually the infections with the worms might be protective against metabolic dysfunctions. And this has also been reported in other settings, in other countries with other type of helminths, and slightly different experimental settings. For example, in India, same thing has been observed. So people infected with salt-transmitted helminths have a lower, have a worsened metabolic profile, and as soon as you deworm them. So, and we also have done this, so this previous study that I just introduced are focused on glucose homeostasis, but this is the same for the lipids. We have also observed in different trials, including one that we have recently conducted in Gabon, that people infected with helminths in these conditions, as hematobium helminths, have a better lipid profile. And then if you deworm them, then they will have a worsening of their metabolic homeostasis or lipid homeostasis. So suggesting again that helminth infections might be beneficial for preventing metabolic dysfunctions. But how it works? I mean, all these human studies are mostly descriptive, and it's difficult to go deep into the mechanism. So we have to rely on other type of models, and these other type of models are usually rodents. So, and it has been also shown that in rodents infected with infections with helminths, different type of helminths, are actually inducing a type 2 immune response in metabolic organs, and this is associated with improvement in metabolic homeostasis. And at LUMC, we have mostly worked with Schistosoma vansoni, and yeah, 5 years ago, we have published our first papers trying to understand better the mechanism by which helminths might actually promote a metabolic homeostasis. And this is the work of one of my previous PhD students, Leonie Hussards. And what she has found is that if you put mice on high-fat diet, they are becoming obese. And if you infect them with S. mansoni, then you will find a very strong increase in all these prototypical type 2 immune cells in metabolic organs. So Th2 cells, eosinophils, and alternatively activated macrophages. And this was associated with an improvement in whole body insulin sensitivity and glucose tolerance. But I mean, you can always postulate that putting a big worms in the small mice, I mean, might actually have a lot of different side effects, and that might explain this improvement in whole body metabolic homeostasis. But interestingly, we could phenocopy this effect by just injecting mixtures of molecules that were isolated from the eggs of these worms. So suggesting that actually some of these molecules are actually inducing this type 2 immune response, and therefore this metabolic homeostasis, this improvement in metabolic homeostasis. So the next question for us was actually to try to see whether this type 2 immune response is really one of the underlying mechanisms involved in this beneficial effect of amines molecules on metabolic homeostasis. And the second question for us was actually to know whether there might be some single molecules in these large mixtures of antigens that could do the job. So to answer the first questions, we start a collaboration with the group of Dr. Penlock in New York University, and we use mice that are deficient for the transcription factor STAT6. And if you remember, STAT6 is a key player in the inductions of alternatively activated macrophages in response to EL4 and binding to the EL4 receptors. So what we did, we put wild-type mice and STAT6 knockout mice on IFA diets, and then we treat these mice with SCA, a mixture of soluble egg antigens, during 4 weeks, twice a week. And we perform a couple of metabolic tests, and also we phenotype the immune cells in the metabolic organs at the end of the experiment. And what we found, and it's just focused on the plain bar now, is that as we have previously found in wild-type mice, there is a very strong increase in EL5 expressing CD4 T cells, so the Th2 cells. There is increase in eosinophils, and there is increase in YM1 expressing alternatively activated macrophages in the adipose tissue of wild-type mice. And this is completely abolished, as expected, in STAT6 knockout mice, which have impaired type 2 immune response. So the model is working well. So what's the impact on the metabolic homeostasis? So we perform a glucose tolerance test, which is actually injection of a bolus of glucose in the mice, and then we follow the blood glucose level in time. And if you see on the left, I mean, the black bars are actually the wild-type lean mice, and the gray is the wild-type obese mice. And you can see that the glucose excretion curve is much higher in the obese mice, indicating glucose intolerance. And if you do the same thing in mice, in obese mice that has been injected with SCA, then you see that there is an improvement in glucose tolerance. And this is depicted by this decrease in the area under the curve. So what's happening in the STAT6 knockout mice? In the STAT6 knockout mice, you can see that the beneficial effect of SCA is completely abolished. It's gone. So what it means is that the type 2 immune response that is associated with SCA injections or treatments is actually contributing to the metabolic homeostasis, to the beneficial effect on metabolic homeostasis. And this is summarized here. But the question now is, among these hundreds of molecules that are present in this SCA mix, could we identify some single molecules that can do exactly the same job? And the idea behind was to eventually have some new therapeutics eventually, or drugs actually to build on to modulate meta-inflammation in obese individuals. And so what we did is actually we start from a kind of educated guess, because in our group for a long time we are working on molecules produced by schistosoma mannsoni that is called omega-1. This is a T2 RNase that has been found in SCA, and it is also released from these eggs. And it has been shown to trigger a pretty strong type 2 immune response driven by priming of dendritic cells. So that's the work of one of our colleagues at the Department of Parasitology, Bart Everts. And we start to work with these molecules. But these molecules are very difficult to isolate from SCA. So what we did, we actually produce recombinant molecules using a tobacco plants. And why we did this, why we use these very specific models, is because tobacco plants can be engineered to make the glycans, so these carbohydrate structures that you see on the right upper part of the figures, which are attached to the molecules. And these carbohydrate structures are key players in the effect of these molecules, because it can recognize some specific receptors, especially on immune cells. And so we want to use these plant-produced molecules to be as close as possible to the native proteins. And then we did exactly the same kind of test. So we inject these molecules during 4 weeks in obese mice, And the first thing we did is to see whether it's also inducing a very strong type 2 immune response in the white adipose tissue. And as we have observed with SEA, we found strictly the same. So increasing Th2 cells, increasing eosinophils, and increasing alternatively activated macrophages. So, and this effect, as expected again, is completely gone if we use STAT6 knockout mice that cannot mount a proper type 2 immune response. So, but what's the effect on the metabolic homeostasis? Well, I mean, we are very happy to see that omega-1 very significantly improved metabolic homeostasis. Even it can restore the insulin sensitivity to more or less the level of lean mice, although these mice are still obese. So it's a very strong and potent effect. But does these effects depend from this type 2 immune response? Actually, the answer is no. Because if we do the same kind of experiment in STAT6 knockout mice, you can see that there is still a very strong decrease, very strong improvement in metabolic homeostasis in the STAT6 knockout mice. So in these conditions, omega-1 can induce a very strong type 2 immune response, but this is not involved in the beneficial effect on metabolic homeostasis. So what's the mechanism? I mean, by contrast to SEA, omega-1 also induces a very strong and potent decrease in body weight. So the first thing we have thought about is maybe it might affect feeding behavior. So what we did, we put these mice in metabolic cages where we can monitor continuously their food intake and their locomotor activity. And what we found is actually omega-1, so this is these two green lines, so two different glyco variants of omega-1, which one without a Lusix branch and one with a Lusix branch, which is very close to the native one, both induce an inhibition of food intake. And the first things we thought was, okay, this is toxic. But if a compound is toxic, then mice are stopping to have any kind of locomotor activity. They are not moving anymore. And we monitored the locomotor activity, and there was no effect. So this is really something that is occurring independently of inducing some toxicity. So it's a true effect on food intake. And what we also looked is whether this effect is mediated by type 2 immune response. And the answer was clearly not, because this inhibition of food intake was also present in STAT6 knockout mice. So the next question for us was to study the mechanism and that are involved in this inhibition of food intake. So I have, for a matter of time, I have removed a couple of slides here. But the most important thing is to show that this, what I'm going to show you now in the coming two slides, is that actually if we mimic the inhibition of food intake that is induced by omega-1 by doing a kind of pair feeding experiment, so we give to a group of mice exactly the same amount of food that is eaten by mice that have been injected with omega-1, we can actually phenocopy the body weight changes. We can phenocopy the effect on metabolic homeostasis. So that's really a decrease, the decrease in food intake that is induced by omega-1 that is explaining the beneficial effect of metabolic homeostasis. And also for a matter of time, I mean, we have investigated a couple of pathways that are known to regulate feeding behavior via the brain. And one of the most obvious one is the leptin signaling pathways that is dampening the feeding behavior when it's increased. And we, with a different type of experiment, we have excluded this effect. So what we have now, we still have a kind of black boxes. We don't really know how omega-1 modulates metabolic homeostasis, but it's modulate food intake, but it's really via this modulation that there is a beneficial impact on metabolic homeostasis. And back to SEA, we know that SEA does not affect food intake, but induce type 2 immune response and improve metabolic homeostasis. So there is still molecules in SEA, which are not omega-1, that might have beneficial effect on metabolic homeostasis and might be also interesting to identify in the coming years. And so that's what my group is doing. Actually, we have a pipeline of different LMIMS molecules, and we are investigating their their underlying mechanism, both the immune-dependent and independent mechanism by which they can modulate the insulin signaling. So I'm going to stop here now, acknowledging all the people that have been involved in different studies. Here are only the people who have worked on these rodent studies, and you all know that there is a large and long history of strong collaboration with the University of Indonesia and the LUMC in the framework of all these field studies. So they are not named here, but you know that it's a crucial collaboration for our department. And I'm willing to answer some questions also during the discussions with all of you. Thank you for your attention. You have to unmute.
Okay, sorry. So thank you very much, Professor Bruno, for your nice presentation on immunometabolism in obesity and type 2 diabetes. And I'm sure that there will be a lot of questions. So there is already one question for you. Good afternoon, Dr. Bruno. Yeah, my name is Fauzan. There are two questions regarding the effect of helminth infection in the metabolic homeostasis and type 2 immune response. Is there any difference between the type of helminth infection to the degree of the type immune response and its effect on metabolism homeostasis? This is the first question.
Yeah, it's a very good question. Well, maybe I will start answering this one. So it's a very good question. Yes, there is certainly some difference, and the type of immune response is that is induced by helminths is species specific. In our group, we have mostly worked with schistosoma mannsoni, but there are other groups that have worked in rodents. That has worked with other type of helminths. Including also some rodent-specific helminths. The effect that is induced might be slightly different, and especially, the antigens that are driving this immune response might also be different. So it's indeed important to work with different type of helminths, and I mentioned in my last slide that the core of the core business of my group is to identify helminth-derived molecules that have metabolic activity, and so we are working with different type of helminth species to try to enlarge this portfolio of active molecules. And indeed, we postulated that depending on the helminth species, there might be some very specific different type of molecules that might do the job.
So why you name it as omega-1, and all this derived from helminth or maybe from some plants? You mentioned also some plant.
Yeah, you can indeed think about molecules that are not derived from helminths, but that are derived from other type of system that might induce some specific immune response that could be beneficial. The plant system is actually a sort of factory to make a large amount of endotoxin-free proteins where we can also manipulate the carbohydrate structures that are present on these molecules and try to mimic the very specific carbohydrate structures that are present on helminth glycoproteins. Because what I did not really mention, and that was maybe a bit unclear, is that among these mixtures, these last mixtures of molecules that are produced by helminths inside the host, there is a large bunch of them that are unique glycoproteins. So they are carrying glycan structures that does not exist in humans. So then that might also explain why there is this very peculiar immune response that in some contexts might be beneficial.
Okay, thank you. Maybe there is a question from Dr. Christian. So it is about the obesity in lean person. Sometimes lean person there is also insulin resistance. So you can explain about the type of immune response. You mentioned about type 1, type 2 immune response.
Yeah, that's a good point. Indeed, I mean, there is two things. There are lean, skinny people that could be insulin resistant and develop type 2 diabetes, and they might be obese people which are perfectly healthy. So, and indeed, that might actually be secondary to a sort of very specific immune response that is occurring in these people. So you might imagine that some of this immune response might be triggered not necessarily by influx of fat, for example, as I mentioned initially. I mean, one of the inducer of this pro-inflammatory environment is high lipid influx in metabolic organs. But in some people, that might be something else. That might be some type of infections. Dr. Farid mentioned the gut microbiota. That might be also some specific metabolites that are derived from the gut microbiota. People remain lean, but this specific local, let's say, disturbance of the gut bacterial species might actually trigger a detrimental immune response in some organs, and therefore leads to insulin resistance. But this is something that is not very well strongly established now. That's just, I mean, some hypothesis that I'm sharing with you. But indeed, it's really important, and I know that there are studies in humans that try to decipher the immune response between the healthy obese and the pathological obese, and that might help us actually to understand which are these tiny changes that might be detrimental.
Okay, thank you. So not all individual insulin sensitive. You want to say that someone also have insulin resistance, yeah? Okay. Yeah. There is also a question from Muhammad Aprianto. So he asking about the relation between obesity and COVID. You know that in COVID there is an increase of inflammation. So maybe you can explain a little bit about the association between change of immunity and also obesity and COVID.
Yes. Dicky, do you want to say something, or do you want me to answer?
Maybe from clinical point of view, as we already did some meta-analysis as well with Alice, one of the students from FKUI, that show that indeed obesity is independently associated with severe COVID infections, and also we are currently working with the McMaster University as well, trying to make a recommendation to WHO about the obesity as one of the independent risk factor. And what we saw so far from our first analysis, we saw that increasing obesity, so that increasing risk of mechanical ventilations, severe COVID infection, also mortality as well. So I think the mechanism is not only related with the immune system. Maybe Bruno will later on discuss about the immune system, but also with other factors as well. For example, about the respiratory capacity, and also other factors that might influence the morbidity and mortality. And also most of obese people have a comorbidity as well, although in our analysis actually it's independent from other comorbidity as well. From immune system, maybe Bruno, you can explain this better than me.
Yeah, sure. But I'm going to build on what you're just saying. But I mean, that's good to always bring it back in this let's say clinical settings first. So indeed, I mean, as Dicky said, this is clear that there is an association between obesity and the risk of having a severe form of COVID. And this is partly, I mean, this is fresh studies. I mean, we only know that we have only studied that during the last 6 months. But what seems to be clear is that there might be some worsening of the inflammation at the whole body level, and it's also include metabolic organs. And you have heard maybe that I mean, the people that are suffering from this very extreme form of COVID, the one that might lead to death, actually, this is actually associated with a cytokine storm. And some of these cytokines, especially IL-6, might be produced in a very high amounts by metabolic organs or the cells that are present in the metabolic organs. What Dicky said is that there is some field studies that have started to work on this. We are also doing some preclinical work in rodents, and we have recently received at LUMC a mouse model that express transgenic mouse model that express the ACA2 receptors that can allow the virus to enter the cells. And what we are going to do is to study the effect of obesity on the immune metabolic response in the organs during infection with COVID. And so we hope that we might decipher some very specific immune response in metabolic organs that can explain this increased risk of having a severe form of COVID. But there are a lot of people working all over the world on this also. So I wouldn't be very surprised that in the coming months, there will be quite a lot of a fundamental study published on this very specific aspect.
Okay, thank you very much. And then maybe there is a question, because you know that this is, in your study, you use also high-fat diet, yeah, Professor Bruno, and also Dr. Farid and also Dr. Dicky, but you on animal model and Dr. Dicky, Dr. Farid in human. So my question is, can we use high-fat diet as a diagnostic tools or prognostic tools for human, for Dr. Farid and for Dr. Dicky?
You mean some of the, you mean using WhatsApp, the information that we are generating in rodents as a kind of future diagnostic tools in humans? That's your idea behind, or?
No, I asked Dr. Farid and Dr. Dicky because they did the study on human already.
Is it about the use of short-term high-fat, high-calorie diet to induce the insulin resistance in the subject? Is that what you meant?
Yeah. It is like OGTT, oral glucose tolerant test. Yeah. So, but in this time you use a fat diet. So, can we use as a diagnostic tools, or maybe it is just for research?
I think, in my opinion, it's this more for research purpose, because yeah, maybe we could induce the certain immune response from a subject that we give high-fat, high-calorie diet, but afterward we get confused about the result that we observe from those subjects. And also, I don't know if that's applied in larger population, whether it will be ethical or not, because we still don't know if those persons already in increased risk of type 2 diabetes, and we give them high-fat diet, although only for short term, like only 5 days, but we still don't know how is the effect on those subjects. That's my opinion. Maybe Dicky could add some more.
Yeah, I think I agree with what Dr. Farid has told us. And also it's technically a bit difficult because it's a 5-day high-fat, high-calorie diet, of which all subjects will be given like extra 1,500 extra calorie of saturated fat. And it's really uncomfortable for the subject itself, because they need to eat that exactly like a whipped cream actually, just like eat the raw whipped cream, and it's really uncomfortable. I know that the OGTT also uncomfortable, but it's only once in a while. But for high-fat diet, you need to be taken like 3 times daily for 5 consecutive days. So for technical, I think for now it's still for research purpose, Prof. Pradana. Thank you.
But actually, conceptually speaking, I mean, you can actually have one acute high-fat diet ingestion and follow the lipid in blood. So that might be like an OGTT, and people can actually react differently, and that might work as a kind of let's say discriminating factors between people that are more prone to develop obesity in the futures and the other. So maybe, I mean, that's something that might be explored.
Thank you.
Not cute.
Okay. Maybe there is also a question here related with the microbiome, gut microbiome. Maybe Dr. Dicky or Dr. Farid can answer the question related to the insulin resistance. So what we can use this information that actually the gut microbiome will influence the insulin resistance. So what we can do?
Yeah, I will try to answer it. There are a lot of studies in the last decades that study this gut microbiome, either the composition itself or the metabolomics regarding the insulin resistance and type 2 diabetes. And those studies show that a lot of varieties that observe between each studies about the gut microbiome, the composition itself, and also the metabolomic or the metagenomics. Although some research say that most of them say that there are decrease of diversity and richness in the subject with type 2 diabetes, and the relative abundance of certain bacteria that produce butyrate. Butyrate is well known as the substrate that could interact with the immune system and induce an anti-inflammatory phenotype of the immune system. And with the less amount of butyrate producing bacteria in the type 2 diabetes, maybe that's associated with the increase of low-grade chronic inflammation in those subjects, and that in the long term, it will increase the susceptibility toward insulin resistance and type 2 diabetes.
Okay, Dr. Dicky?
Yeah, I think almost all have already been explained by Dr. Farid, but we have also research that currently analyzing, comparing the microbiome composition between normal people, normal glycemic, pre-diabetes and diabetes, and hopefully to get the result in the next few months. And about the gut microbiomes also have a close relation with oral microbiome as well. For the oral microbiome, we have already had the data and just submitted for publication, and we saw there is a difference in terms of composition between people with normal glycemia, pre-diabetes, and diabetes as well.
Okay.
Thank you, Prof.
Yeah, thank you. Maybe there is a last question for three of you. So this is a question from our colleague that participate in the clinical immunology module minor, half minor in LUMC. So the question is about the metabolically healthy obese. And some studies reported that MHO has a fewer visceral fat and more insulin sensitivity compared to the metabolically unhealthy obese. So is there any study comparing and explaining two subsets regarding the immune profile? Maybe first, Professor Bruno.
I mean, yeah, there are studies. There are studies that are definitely focusing on this, on trying to decipher the difference between the healthy obese and the particular obesity in terms of immune response in different metabolic organs. What I know is that there are few. I mean, at least in the wide adipose tissue, because I mean the thing is that we cannot access. I mean, some really important metabolic organs like the liver or the brain or the pancreas or even the intestine. But I mean, mostly the tissue that has been mostly studied is the wide adipose tissue, and there might be some slight difference in terms of immune cell populations. And one of the recent cell type that has been shown to be to maybe play a role, it's called mate cells. and they are differently expressed in healthy obese or let's say pathological obese. So that's something that is currently explored. I know that there are some French colleagues that are working on this at the moment, but also the new technology that has been developed recently. So this single cell RNA sequencing, I mean, might actually bring some new insights and identify very specific new type of new subsets of cells that might have a pathological roles. Yeah, in the coming years, there is certainly new insight that will allow to better understand the difference between these two populations of people.
Thank you, Professor Bruno. Maybe Dr. Dicky?
Yeah, thank you, Professor Pradana. Yeah, I think the idea of a metabolic healthy obese is quite, controversial, I suppose, because I think previously we divided between metabolic healthy and unhealthy, but later on, actually, this metabolically healthy is actually not really healthy, but then maybe in the next 5 years or 10 years, then we develop unhealthy profile as well. But still, the question is why one group of obese people currently not developing the metabolic abnormalities, but at the same time compared to the other people with the same set of obesity. And Dr. Atika is currently now writing a proposal trying to see the role of the gut permeability as well, comparing the gut permeability between those people with obese, comparing those with metabolic abnormality and normal healthy obese. And then later on, we can also compare the gut microbiome between those groups as well. Maybe later on, we can also collaborate with Bruno as well. It would be interesting to see the immune cells in both groups as well. I think that's all from me, Dr. Pradana. Thank you.
Thank you. Dr. Farid?
Yeah, I just want to add some little things that for the definition of the metabolic health itself, it's kind of not really a good definition right now. The definition that proposed, what I know from the latest publication in this 2020 is from JAMA, if I'm not mistaken. They similar like the definition of the metabolic syndrome. So the people that are the subject without the indices of the metabolic syndrome criteria, although for the blood pressures, they use the cutoff of 120 and for the systole and 80 for the diastole. So if the indices are still in the normal limit, they called metabolically healthy. So that's the definition, but we still don't know in the future if this definition would be used or not. That's one thing. And then regarding the immune system, yeah, I've read a publication regarding the mucosal associated T cells that associated with the obese and insulin resistance. I forgot in which journal, but that's very interesting publications.
Thank you very much, Dr. Farid. I think I have to close this very interesting session. You know that recently obesity is, alarm us because he already mentioned that obesity is one of the risk factors not only for diabetes and cardiovascular disease, but also for the severity of COVID-19 and also the cause of death. So I think we have to aware about the pathophysiology of obesity. Don't think about the healthy obese, but the obesity itself is already a risk factor for COVID-19. So I want to say thank you very much for all three speakers, Professor Bruno, Dr. Dicky, and Dr. Farid, for your very important contribution on this guest lecture. And also thank you very much for the all participants that you already follow this lecture quite well, and there are a lot of questions, and I'm sure it will be beneficial for us. And thank you very much once again. I return to the MC. Thank you very much.
Thank you, Bruno.
Thank you.
Terima kasih, Pradana. Thank you,
Pleasure to be
Pradana.
there.
Thank you.
Terima kasih, Farid. Thank you. Bye. Makasih ya Farid.
Sama-sama Bang, terima kasih.
Makasih Mbak Indah, makasih semua partisipan, makasih.
Ya, terima kasih.
Ali.
Ya, in live ya, makasih.
Ya, silakan.
Tidak, Mbak.
Ya, silakan. Terima kasih.